Very pleased today to introduce Dr. Day. Sharlene Day is here with us from the University of Pennsylvania. She, by way of introduction, did her bachelor's degree at MIT and then her medical school training at New York University. Then she went to Michigan for additional training, residency, fellowship, postdoctoral training, before joining the faculty there, and has conducted an excellent both clinical and research programs. Was at Michigan as an assistant professor, associate professor, and then was recruited about 3 years ago. Is that right? The University of Pennsylvania. She, as we'll learn in her talk, does excellent both clinical and basic research in hypertrophic cardiomyopathy. She's a real leader and is active with NIH study section. She chairs the Therapeutic Development and Preclinical Studies study section, which is a great section. It's one of the relatively new ones that we're just starting to learn more. With that, she's going to speak today about emerging therapies for hypertrophic cardiomyopathy, some of the old and new things. And we'll turn it over to you. Thank you.
Thank you so much. The introduction and the hospitality here has been wonderful. Had my own little desk, plants. It's been wonderful. It's been really smooth. And Diane has definitely made this day go very easily. And I've been really enjoying meeting with people so far, and I'm looking forward to more meetings this afternoon and dinner tonight. So I have this talk structured in a way that's a little bit more patient oriented than maybe some of the other speakers that you have here. And I thought it might be a little bit more broader interest because, many of you are working on things that you want to translate to the clinic. And I think a lot of the sort of concepts that I'll talk about today with hypertrophic cardiomyopathy apply to other forms of cardiomyopathies and heart failure and disease processes. And there are some things that we can learn from the past and some new drugs and other interventions that I will share with you. So here are my disclosures. The only relevant one really is Bristol Myers Squibb, because I am going to talk about Mavacamten today, which is a new drug that was just FDA approved. So I always start with my acknowledgements because I am so grateful to be part of this huge network of people across the globe. These are people who I work with in patient-oriented research, from those at Michigan, who I'm still very close to and go back frequently, in faculty. When I joined, I was the third, and now there are 6 primary faculty who see patients in the inherited cardiac disease clinic. We have several genetic counselors, surgeon, interventionalists, nurse practitioners, nurses, and research coordinators, program managers. I don't know if I forgot anybody, but it's a growing group, and it's really a just such a pleasure to work with everyone. And then I'm also part of several collaborations and registries. For example, the SHARE registry has been something that we've been working on for a long time. We're in our 10th year now. And here's a picture of us at my house, actually in Philadelphia, when AHA was in Philly before the pandemic hit, and we barely had any furniture in the house, but we had people over anyway. And then there's a couple of exercise studies that I'll touch on as well. One of them, the RESET-HCM study. Just to tell you a little bit about the SHARE registry, because I'm going to show you some data from the registry. So this was a multi-center and international effort where we've now put together over 10,000 patients with hypertrophic cardiomyopathy, very detailed genotyping clinical outcomes, and phenotypic features. Many of them are sarcomere positive, and we have genetic testing information on many. We're now expanding to genetic dilated cardiomyopathy as well. And then I'm very indebted to my lab folks. So these are pictures that span from March of 2020, literally the week before everything shut down when we were in Keystone together. I think this was actually in Keystone. It wasn't at Snowbird. And then, there was kind of a gap in our social activities for a while, and now we're kind of back together again. And this is a picture from Michigan celebrating a thesis defense of an MSTP student in Michigan who continues to work with me and is now moving on to his clinical years. And I'm very grateful for the people that moved with me from Michigan, people who've stayed and continue to work with me, and others that have joined the lab since. And then patients, you know, a lot of what I do is very much inspired by the patients I see, and I'm, you know, I'm really just in awe of what they're able to accomplish with all the challenges and the tragedies that many of them have suffered. And so it's very inspiring, and it definitely, you know, drives a lot of my motivation for what I do. Okay, so just to give you a very broad overview of hypertrophic cardiomyopathy, this is the most common genetic heart condition. It's defined somewhat simply as unexplained hypertrophy at or exceeding 15 millimeters in any wall segment when normal is about 10. Although I'll tell you that that definition is really pretty vague and doesn't really encompass the types of patients that we see in trying to distinguish between HCM and other forms of hypertrophy. But this disease has a huge variation in terms of the age of onset and disease severity, anywhere from early, you know, childhood years to later on, you know, later middle to adult years. And the disease severity varies widely. We have some patients with no or mild symptoms who have excellent quality of life, normal longevity, whereas others suffer a variety of complications, including heart failure and arrhythmias. HCM also looks a lot different in different people. And so these are some cardiac MRIs, these three just showing a few different morphologies. The most typical is the asymmetric septal morphology, where let's see if my picture is going to work here. Do you see this up here? You do. Okay. So here, and this is the MRI showing the left ventricle. I'm showing the hypertrophied segment here being the septum. This is an apical variant where the hypertrophy is confined to the apical segments. And then there's another variant here where the hypertrophy is mostly in the mid cavity, and that those patients are predisposed to developing an apical aneurysm. And then outflow tract obstruction is unique physiology to this disease, where the hypertrophied septum sticks out into the outflow tract and redirects the blood flow around so that it drags the mitral valve leaflets in the outflow tract and causes resistance to blood flow as it's exiting the heart, and that causes pressure buildup inside the LV. And a lot of patients get very symptomatic from that. So that's important because there are a subset of those patients who will go on to have surgery, and we use a lot of that tissue from the septal myectomy for discovery of disease mechanisms, and I'll show you a little bit of that later. So just to give you a flavor for what this disease is like in terms of its disease trajectory. So many patients with HCM really do have a substantial disease burden. The mortality is about 3 to 4-fold higher in patients with HCM compared to the general population across all age strata. And they also have a very high cumulative risk of adverse outcomes. So these are cumulative incidence plots that are stratified by age of diagnosis for those under 40, middle age 40 to 60 and over 60. And so there's a few things you can learn from this. One is obviously the earlier you're exposed and the earlier you develop it, the worse you do over time, and such that by the time you get to 70, if you're diagnosed as a young adult or a child, you're likely to have suffered at least one of many serious complications. If you're older, you know, then you may have, you know, then only maybe about 25% of those patients will develop complications. And there's also a long lag time. So you can see like the curve's flat for a while before it starts to go up. So a lot of times patients may be diagnosed incidentally, for example, based on abnormal EKG or screening because of family members that have HCM, but maybe they won't develop any symptoms until they're, you know, 20, 30 years down the road. So just breaking that one cumulative incidence plot, which is sort of a composite, into 3 individual composites. When we look at ventricular arrhythmias that can cause sudden death, those are actually much less common than heart failure atrial fibrillation, although it's one thing HCM is kind of known for, and the most feared complication because it's sudden and unexplained. But those who are diagnosed under the age of 40 have about a 35% chance that they'll develop a serious ventricular arrhythmia in the course of their lifetime. Those over 60, it's less than 1%. So that definitely plays into decisions around implantable defibrillators for primary prevention. And then heart failure and atrial fibrillation are by far the most common complications. And about 25% of patients with HCM have developed atrial fibrillation or heart failure. So to give you a little bit of background on genetics, because I think it's important when we think about therapeutics, and more going, you know, as we start to move towards, you know, gene targeted therapies or gene specific therapies, I think we have to understand a lot about the genetic basis. And so it's been 32 years since Cricket and John Seidman pioneered the field by discovering the first genetic mutation that causes HCM, which is a mutation in myosin, RH403 glutamic acid. And this they discovered through linkage analysis of a very large family. And now where we are is that we understand that the sarcomere is really the primary genetic etiology of HCM. If you look at this pie chart, myosin binding protein C and myosin lead the way in terms of the percentage of cases that are explained by mutations in those genes. And then the other 6 sarcomere genes make up a smaller percentage. There's about 5 genes outside of the sarcomere that have very strong evidence for disease causation in HCM. The most notable thing about this pie chart is the gene elusive group, which is this big gray box. And it hasn't really, it's probably maybe now 50% since this was published 5 years ago, but we haven't made a lot of inroads into, identifying more genes that are inherited in Mendelian fashion with a strong effect. But what we have learned is that HCM is not just a Mendelian genetic disease, but it's also a complex trait. And a couple of GWAS studies published in Nature Genetics back in 2021 identified a number of risk alleles for HCM. And one of those looked at alleles in both HCM and DCM and showed that if it's a risk allele for HCM, it was a protective allele for DCM for most of these. And so I think that's really interesting observation and suggests that perhaps a lot of these risk alleles are in some way regulating contractility. But when they put all these together and created a polygenic risk score, for those individuals that are in the upper 20% of that risk score, they had about a 2 to 2.5-fold likelihood of having HCM. And if they were in the lower 20%, they were about half as likely to have HCM. And the other thing that came out of these studies, which is something I think we've, practicing in this field for a long time, recognize that there are a lot of acquired traits that contribute to this disease. So in this graph on the right, they're looking at hypertension, systolic blood pressure, diastolic blood pressure, body mass index, and type 2 diabetes as acquired traits. And in those that are sarcomere negative in blue, particularly diastolic blood pressure confers about a 4-fold risk of having HCM. Body mass index as well, diabetes was kind of borderline, but definitely blood pressure and body mass index. And even in those who carried a sarcomeric gene mutation, there was still an influence of these traits, suggesting that that these are genetic modifying factors and acquired modifying factors, even for those who have sarcomeric disease. So I think we're learning a lot more about the genetic complexity of HCM. I think to sort of summarize everything on this slide, about probably 40% of HCM, 40 to 50% is explained by Mendelian disease. Most of those are sarcomeric disease. There are probably some that we haven't discovered, and certainly in non-European ancestry populations, we still have a lot to work to do in genetic discovery. But there's probably about 40% that we can now call non-familial HCM. They don't have any family members diagnosed with it. So these are isolated cases in which probably polygenic and acquired factors are causal. And these proportions will vary depending on the referral population, where you're studying these patients and where they're coming from, and also their ancestry. And we still have a lot of work to do in non-European ancestry individuals, because most of these data come from Europe and the US. So I think that's an important background to understand when you're thinking about the therapeutic landscape of HCM. And here again, I'm talking about HCM, but you could probably substitute in a lot of different cardiomyopathies or other forms of heart failure in this type of a landscape where you think about an inciting factor. In this case, it's something genetic. So it's either a single coding variant that has a strong effect and maybe combined with or substituted for other multiple variants. And so an allogenic or polygenic trigger for disease. And that these genetic variants cause some primary effects, whether it's loss of function of that particular gene or a gain of function. And then that leads to some secondary effect, changes in biophysical properties, altered protein-protein interactions. And then we do a lot of hand waving because we don't really know how the heart develops hypertrophy and fibrosis and all of the phenotypic features that we're familiar with that manifest in patients. And once they develop disease, then of course they're susceptible to complications. So when you think about where we are in terms of current therapeutics, we're really on the far right side of this, right? We're really treating patients not only once they have disease, but once they've developed complications. And so we can use medications to treat symptoms. We have surgery to treat obstructive patients that are symptomatic, who are refractory to medical therapy. heart transplant for those that obviously have very advanced heart failure. And we can prevent arrhythmias or at least treat them by either radiofrequency ablation or implantable defibrillators. But we really don't have anything right now that we're targeting to patients who may have the disease who are not symptomatic yet, or who maybe haven't developed yet as preventive therapy. And that's clearly where we want to go. So I think working backwards, we can think about modulators of secondary remodeling. There are a number of pathways. I'm just naming a few here. Metabolism and energetics, I know is of interest to this group, and I'm going to show you some data in that we're area that we're working on, and calcium signaling that I'm not going to talk about today, but obviously that's very important. Neurohormonal pathways and lifestyle interventions that I think, can modulate some of these acquired factors. We can go further back and think about modulators of primary disease pathways, things that might stabilize a protein or either inhibit or activate a protein-protein interaction that's lost. These are, I think, further not as well developed at this point, and there's not really anything other than the myosin modulators that I'm going to talk about that could potentially be in this pathway. And then genetic therapies, gene replacement, gene silencing, somatic gene editing. I think, there's obviously a lot of excitement around this and an enormous number of challenges as well in implementing, but I think it is, it's here, and we're starting to see that at least in diseases that have very severe, high penetrant and severe disease like DMD and others. So I'm not going to talk about that today, but I'm happy to entertain questions if anyone wants to discuss later. So I'm going to just cover a few different stories, and I'm going to stay mostly on the right side and things that have either are in current, in current practice and things that we're using now. Or things that we're developing that I think are close to maybe translation to clinical therapy. So the first story I'm going to tell is about something new. And I don't know how many people in this room have heard about these myosin modulators or myosin inhibitors or activators. Obviously, there's a few clinicians in the audience who are familiar with them, but I'm going to just introduce them. So these are a class of drugs that were designed to decrease power output by decreasing myosin ATPase rate. So they were developed on the premise that an increase in sarcomeric power and contractility is a primary molecular cause of HCM. And that is really what we see: increased ejection fraction, for lack of a better measurement tool to measure contractility. And so a hyperdynamic state is something that's felt to be fundamentally, at the core of the molecular cause of HCM. And so, initially, a company, Myocardia, developed a small molecule screen to screen for compounds that bind myosin and reduce ATPase rate. And so you can reduce force by a few different ways. So the ensemble force is generated by the force that's generated by a single myosin, multiplied by the duty ratio, which is the amount of time that myosin and actin spend bound to each other within the cardiac cycle, and then the number of heads that are available to bind to a number of myosin heads that are available to bind to actin. And so the way that Mavacamten works, which is the first drug in this class, is that it stabilizes a conformation of myosin that puts it in a super relaxed state. And you may have heard of the interacting heads motif. That's one of the conformations that can assume a super relaxed state. And in that state, when the heads are kind of folded over on each other, they can't reach out to bind to actin in this state, in the actin-activated state. So that's the way Mavacamten at least works. And that drug was developed by Myocardia, which was bought out by Bristol Myers Squibb, has been through a phase 3 trial. I'll show you in the next slide, and was approved by the FDA on April 28th. And we are prescribing it. It's under a risk mitigation strategy through the FDA. So it's pretty complicated, and it's limited number of sites that are able to prescribe it. It's a lot of work. Anyway, we'll get there. Aficamten is the next in class. It was developed by Cytokinetics, and that's currently in a phase 3 trial. So this is the pivotal trial. This is the Explorer trial that led to the FDA approval. In this trial, there were 250 patients with obstructive HCM who were randomized, and it was a placebo-controlled trial. And here I'm just showing you a couple of the major endpoints. One is that Mavacamten, most patients reduced outflow tract gradients pretty substantially compared to placebo. And then their primary endpoint was a combination of peak VO2, which is a change in exercise capacity, and New York Association class, which is assessment by the provider of how limited they are in terms of like shortness of breath with activity. And so the primary endpoint was met in 37% of patients on Mavacamten versus 17% in placebo. And if you can see in this graph, the patients on Mavacamten, all of them were symptomatic at baseline, and 50% became asymptomatic by the end of the trial. And on placebo, 20% became asymptomatic at the end of the trial. So this is what, again, led to the approval for obstructive HCM. However, there are a lot of patients that have symptoms with hypertrophic cardiomyopathy that do not have outflow tract obstruction. Presumably that's on the basis of diastolic dysfunction. But there may be other things as well that contribute to their symptoms. And there was a small phase 2 trial of Mavacamten non-obstructive HCM that did not meet their primary endpoint. There was no difference in peak VO2 or NYHA class between the groups at the end of the trial at 24 weeks. There was, however, a signal potential for efficacy in that there was a drop in a biomarker called NT-proBNP, which is a marker of stretch and strain in the left ventricle. And so it was about a 50% reduction in the patients on Mavacamten. So there is another trial planned, and it will be underway shortly this fall, a large trial where they're planning to enroll over 400 patients with non-obstructive HCM. So we'll see how that turns out. That's going to take a little while to enroll. And there is potential for this class of drugs to really have benefit beyond just reducing obstruction and symptoms. I think, based on the studies from mice, this is from the Seidman lab, if Mavacamten was given in an early stage before the mice developed any hypertrophy or fibrosis, then the hypertrophy and fibrosis. So here you can see 3 different mouse models, Mavacamten's in blue, and you can see that there was less wall thickening that developed over time. And this is fibrosis, where you see that there's less fibrosis. So I think there's potential, but we're not quite there yet in patients. So here's where we are currently for symptomatic obstructive HCM. Mavacamten is available for clinical use. There's others in the pipeline, including aficamten. And then, we still don't quite know which features are going to predict treatment response. It's certainly not universal. We've had a number of patients who really don't seem to respond well at all. And so we don't really have a handle on that yet. For non-obstructive, the jury is still out, and we're not sure which subsets of patients will glean benefit. But there's certainly potential benefit in terms of improving diastolic filling that these drugs, will hopefully benefit some patients with non-obstructive disease. And then for the asymptomatic early stage, there's hypothetical benefit, but we're not currently there in terms of testing. So that's the newest thing in HCM. So that's something that I always get asked to talk about, because a lot of people are very excited about it. And it's really nice to have the first drug that was ever developed to treat HCM specifically, and in something new, a new option to offer patients. So my next story is something old, which is repurposing of an old drug, an angiotensin receptor blocker in this case, to treat HCM. So these are drugs, if you are familiar with angiotensin receptor blockers, Valsartan, Candesartan. These are drugs that are used to treat hypertension, to treat cardiomyopathies of mostly reduced ejection fraction. And they also have other properties in that they antagonize molecules like transforming growth factor beta, which can be important in development of cardiomyopathy. So in this case, this was a really nice example of a story that translated from a mouse study directly to humans in a relatively short period of time. I guess 12 years is pretty good. Where this was data from the Seidman lab, in which they had discovered TGF-beta as an important molecule that was upregulated early in the pre-hypertrophic stage of their mouse model. And when they antagonized it with neutralizing antibodies, that prevented the hypertrophy and fibrosis. And then they used the angiotensin receptor blocker, Valsartan, and see all the same effect. And so the idea behind the clinical trial called VANISH was that Valsartan lessens hypertrophy and fibrosis if given at a young age. And so initially we were going to give it before the onset of hypertrophy in patients. So the so-called genotype positive, phenotype negative. And we do have a cohort of those patients we gave it to, but we decided that it's sort of tough in a 2-year time period to show any benefit, because many of those patients will look exactly, they're not even patients, right? They don't have the disease yet. So we decided instead to target early stage disease. So patients that had some hypertrophy, but were very young. So our average age was 22. So we had a lot of pediatric patients and young adults in the study, who in vast majority were completely asymptomatic. So, and what we did in terms of a primary endpoint was a composite endpoint that I'm showing you here in this forest plot, where we looked at the change to composite Z-score among 9 different measures of structure function. We included BNP and troponin I, so things so biomarkers as well as diastolic function, systolic function, wall thickness, chamber size, et cetera, and measures of diastolic function. And so what we saw was that over 2 years, the patients that were on Valsartan had a beneficial effect compared to placebo when you looked at all of these 9 scores and 9 measures that we put into a composite score. And all of the subgroup analyses, with the exception of those with the greatest wall thickness, seemed to benefit. So, so we were really encouraged by that. If you look at just a couple of examples of individual measures, so left ventricular end diastolic volume, for example, improved on Valsartan, which suggests that there's, positive remodeling compared to placebo, where it didn't change. And then for a measure of diastolic function, we saw that declined over the 2 years on placebo, where it stabilized with Valsartan. So we saw both some reversal and stability with Valsartan, and we're now able to offer this to young patients with early stage HCM. And my experience so far has been that they're very excited to do this, because they want to, anything that they can do to prevent their disease from potentially progressing. And the nice thing about Valsartan is side effect profile is great. Patients tolerated high doses of Valsartan despite being normotensive. Really, I've had very few not being able to tolerate it. So, so this is exciting. So something old, again, being repurposed for HCM. So the next story I'm going to tell is about, is sort of an old story, but one we haven't solved yet. And that's an impairment in cardiac energetics. And this is certainly true of not just HCM, but a number of other cardiomyopathies that lead to heart failure, where we know that cardiac energetics is impaired and really could be a very viable target for therapy if we can figure out exactly what to target. So these are data from almost 20 years ago using P31 MR spectroscopy in patients with HCM. And here you can see that this is a ratio of phosphocreatine to ATP. This was reduced in patients with HCM compared to controls. And even in those that didn't have LVH yet, so these are just carriers, suggesting that energetics is impaired in a much earlier stage before the disease even develops. And there have been a couple of attempts to try and improve cardiac energetics in patients with HCM. Both trials have failed. One was with perhexylene, which is an inhibitor of fatty acid oxidation, with the idea that it would shift to glucose oxidation. But the perhexylene maybe showed some potential benefit, but it's also very toxic and just couldn't advance through clinical trials. And this is an example of a trial of trimetazidine, which is another fatty acid oxidation inhibitor, which actually showed potential detriment. Even after 3 months, there was a decrease in exercise capacity and 6-minute walk distance. So, I think it's very clear that this is in the impaired cardiac energetics is really important and fundamentally important in terms of the pathophysiology, but I think we're sort of barking up the wrong tree. So, so this was something I was very interested in when I moved to Penn, is that I really didn't have anybody in my immediate sphere, who was doing cardiac metabolism. And then I moved to Penn and I had Dan Kelly and Zoltarani and Ken Margulies around me. And so I'm like ensconced in cardiac metabolism. And we had already collected a lot of human heart samples at Michigan from non-failing donors, and then from patients who'd had septal myectomies. And we knew what their genotypes were for most of the samples. And so what we did was we had done a whole heart proteomic study in collaboration with Mike Previs at the University of Vermont. And I'll show you those results on the next slide. But that really prompted us to want to look into the metabolomics. And so when I moved to Penn, that was a perfect opportunity. So I'll show you, this is already published, but I'll show you a few pieces of data from this study. So these are the proteomic results in which we compared each one of these groups with HCM, no sarcomeric variants, so genotype negative to those with myosin binding protein C and those with myosin, and then compared them all to non-failing controls. So these plots, these volcano plots, are all compared to, or fold changes compared to the non-failing controls. So the first observation is that there really weren't that many differences between with genotype. So it didn't actually matter what the genotype was. The findings were very much the same. Like, we had very few things that differed. Some of them in the sarcomere group were a little bit more greater magnitude difference than the non-sarcomere, but it was pretty much the same proteins. So overall, we saw an increase in Z-disc proteins, cytoskeletal proteins, those involved in extracellular matrix, of course, other fibroblast proteins. So that was expected and has been shown previously. And what we saw in terms of the downregulation was profound downregulation of mitochondrial genes, those involved in fuel metabolism, particularly those involved in long-chain fatty acid beta oxidation, as well as energy transduction. So there's a 50% reduction in CKMB in the HCM hearts compared to controls. And so then when I came to Penn, we did a targeted metabolomic profiling of the same samples. And there are 100 different metabolites that differed between the groups. Again, we really didn't see any difference with genotype. So I'm just showing you just a sampling of some of these. So there was marked reduction in the availability of ATP in the human HCM hearts, again, sort of similar across all three groups, and near complete absence of phosphocreatine. It was pretty much undetectable in most of the samples compared to non-failing. And we also found that the reduction in availability of ATP was inversely proportional to the left atrial size. And in HCM, the left atrial size is a very sensitive marker of diastolic dysfunction. So the bigger the left atrium, the worse the diastolic function of the LV is. And so the fact that there was an inverse correlation suggested to us that perhaps the reduction in availability of ATP could contribute directly to diastolic dysfunction. Now, we don't know that for sure. We can't, these types of studies, you can't prove causation, but that was at least an interesting thought. We also observed a marked reduction in pretty much all the fatty acids, but particularly in those long chain fatty acids. And I'm just showing you a couple here, the C16 and C18. And there was also an increase in ketones. The 3-hydroxybutyrate is a ketone body that's used by the heart for an alternate fuel, as well as an increase in the only fatty acid actually that increased was the one that's a breakdown product of ketone bodies. So for those of you that may not think about ketones that often, like I didn't before this, but ketones are made in the liver and then they circulate in the bloodstream and get taken up into distal organs, including the heart. And ketone oxidation has been shown to be an adaptive response to metabolic stress. So these are data from Dan Kelly's group, where he showed if they knocked out an enzyme called BDH1, which prevents the interconversion of beta hydroxybutyrate to acetoacetate, this one right here. If he knocked that out, then when they did expose the animals to a combination of thoracic aortic banding and myocardial infarction, that those animals that had knockout to BDH1 did worse, and that they were more dilated and had lower ejection fraction. And then this is work from Zalt Irani's group that they just published in Science 2 years ago, where they took samples, and these are patient samples who were undergoing an ablation for atrial fibrillation, and they sampled the vein, the artery, and the coronary sinus. So they could then look at the metabolites at each part and figure out what the heart was using for fuel. So what was coming in and what was coming out. And what they showed is that those patients that had atrial fibrillation but normal cardiac function, so preserved ejection fraction without heart failure, they use mostly free fatty acids and a little bit of ketones and amino acids. Whereas those that had reduced ejection fraction with heart failure used fewer fatty acids and about 3 times the amount of ketone bodies. So this is felt to be an adaptive response. So when you think about the HCM heart, we know that it's energy starved, and there's been a lot of focus on the consumption side, where we know that there's increased consumption and by the myosin ATPase because you're generating more force. But I think less attention has been paid to the fact that there's decreased availability of energy through a variety of different mechanisms and mitochondrial dysfunction. And in transcriptional remodeling that's down-regulating a lot of the enzymes involved in fatty acid oxidation. So I think in terms of therapeutics, we're trying to target the increased consumption with the myosin modulators. I think that's one potential, avenue, but also thinking about how we can improve the availability of energy for the HCM heart. And again, this applies to other forms of heart failure. So if I take a page, literally crop an image out of a review in targeting cardiac metabolism and heart failure, there were a couple of potential ideas here that would appeal to me in applying to HCM. So one would be to stimulate ketone oxidation. And the other would be to stimulate fatty acid oxidation to try and increase energy production. So in terms of stimulating ketone oxidation, you can do that acutely by infusing ketones or having people drink a ketone ester drink, which is pretty nasty from what I hear, and or going on a ketogenic diet, which has some downsides to it because of the other constituents in there are not particularly heart healthy. So the only pharmacologic way that we know of so far that could stimulate ketone oxidation in the long term are drugs called SGLT2 inhibitors, sodium glucose cotransport inhibitors, which again, if there's clinicians in the room, you've definitely heard of this because this has been a blockbuster drug that was really a huge surprise for heart failure, both in preventing heart failure and also in treating patients with heart failure. And tremendous benefit for those with reduced as well as preserved ejection fraction. And so these, still curious as to how these drugs are working because SGLT2 is not expressed in the heart. It's expressed pretty much exclusively in the kidney and a little bit in the gut. And so there's a lot of, interest in trying to understand what they're doing, but at least one of the mechanisms is that they can stimulate ketone production from the liver. And so that's one mechanism that they may be working to improve energetics. So we are, hopefully, I don't have this absolutely finalized, but we hope to be starting a trial of SGLT2 inhibition in patients with HCM as a multi-center trial. And then the other potential option in terms of stimulating fatty acid oxidation, and I'm not aware that there's any pharmacologic way to do this, but exercise has been shown to do this through signaling through PGC1 alpha that stimulates mitochondrial biogenesis and increases fatty acid oxidation. So I'm going to share with you in the next probably 10-15 minutes or so we'll have time for questions, some work that we're doing in exercise. And this has been an area of interest of mine for a while. As a clinician seeing patients, I spend a lot of time just trying to get people to exercise at all. As you know, this is, sedentary lifestyle is problematic for a lot of Americans. And so with HCM, it's particularly problematic because there's always been this fear about exercise triggering ventricular lethal arrhythmias. And so one of the things that we wanted to do early on when we first started our clinical program was to gather some data around exercise practices in HCM. So we conducted a randomized trial of moderate intensity exercise training. This was not anything fancy. People were exercising at home, walking on a treadmill outside, riding a bike, elliptical, basically sort of stuff you would do at the gym, moderate intensity level. This is not high intensity or anaerobic training. But we really just wanted to get some data because there was a lot of concern about the safety of these types of exercise practices. And so what we found is that our primary endpoint was a change in peak VO2 or exercise capacity over the course of the study, which was 4 months. And we did achieve that. Those in the exercise group had an increase in peak VO2 on average of 6%. And they also had an improvement in their physical functioning as judged by a standardized questionnaire. Importantly, we didn't see any major adverse events, so there were no arrhythmias in these patients. So that was encouraging and has since prompted a much more liberal approach, I would say, to exercise practices in patients with HCM. But we still are getting a pretty modest increase, and we've thought, well, maybe if we could get patients to exercise more at a higher intensity, we could get a bigger bang for our buck. And there are a couple of trials that are near completion that have done some high intensity exercise training in patients with HCM that look very promising. But one of the approaches that we took was to bring this back to the lab and study this in mice. And for a couple of reasons. One is that it was a little bit higher throughput than trying to put patients through. It's like logistically very challenging to do this because you have to do it for the most part in supervised training type protocols. And the other is that we wanted to try and understand whether exercise could actually have an effect on the disease process itself. In other words, is there a disease modifying effect of exercise? And could we identify molecular transducers that may identify pathways that we could target, pharmacologically to amplify the effects? So Jonathan Herrera is an MSTP student at Michigan. He started this project from scratch. He has exercise physiology background, and he's really done a tremendous job in developing this protocol that mice will comply with that produces a good exercise training response. So he basically did this as a 4x4 where the mice get on the treadmill. This is a forced exercise training response on an incline. And then they exercise at 85% of their peak VO2 for 4 minutes and go down to 55% and do that 4 times. And they've tested before and after so that we have their peak VO2 before and after the training. And we did it for 6 weeks. And we broke it into females and males so that because there actually are some important sex differences here. So But we saw good training effect in both females and males. So these are, this is the female data. To orient you, these are non-transgenic sedentary mice, transgenic sedentary mice. And the transgenic sedentary actually decline their peak VO2 over this time, whereas both groups, non-transgenic and transgenic, had an improvement. And the same was true of the males. It was a little bit of a lesser effect in the transgenic, but still we saw a very significant difference between their sedentary counterparts. So if you look at the effect size in terms of what the training effect was, it was about 18% increase in peak VO2 in the non-transgenic animals and a little bit less, but still pretty substantial in the transgenic. So I think he came up with a really great model system. He's also, I'm not showing you all these, but he's done training on a rotor rod and doing acceleration and endurance capacity as adjunct measures in training modalities. So he's really developed some protocols I think will be useful for anyone who is interested in incorporating exercise capacity and testing into their model systems. I can tell you that the high intensity exercise training did not alter any of the phenotypic features of HCM. So here I'm just showing you the baseline echo data, where the LV volumes are lower in the transgenic mice, left atrial volumes are higher, and the ejection fractions are higher. So similar to what we see in patients, and they do have some asymmetric septal hypertrophy, and none of these change with exercise training. These are some post-mortem studies where we're showing here an increase in the left atrial weight. There was a big increase in left atrial weight in transgenic mice, but no difference with exercise training. And the same is true of fibrosis. Everybody's always interested in fibrosis. This model doesn't really have much fibrosis. The males have a little bit, and it's not different. So we haven't done anything detrimental. We didn't think we would probably do anything beneficial because these were pretty late stage. They were a year old and it was a short intervention. But we didn't do anything bad. So that was good. We've been very intrigued by the RNA sequencing data, which we're still analyzing. So I'm going to show you this is somewhat preliminary in terms of how we're displaying it, but I'm very confident in the actual results. But we're still working on the display of it. So first, there were huge sex differences. So we've analyzed the males and females separately. And just to show you here, these are now PCA curves of females on the left and males on the right. In the females, the non-transgenic sedentary and non-transgenic males are sort of about the same. They didn't really have much difference in terms of their transcript expression. HIT training in females, for some reason, did shift about 100 genes. They were almost all increase in transporters, like and ion channels. But that didn't happen in the males. And then the HCM group was really interesting. So the males are just more severely affected in general. They have a lot more differentially expressed transcripts. But the HIT training in females didn't really do anything, whereas it actually had a pretty significant effect in shifting a lot of these genes back. And I'll show you some in the next couple slides. So here's a gene ontology analysis where I show females on the left and males on the right. And these are the pathways that we either upregulated if they're on to the right of the zero line or downregulated to the left. And so females had 854 differentially expressed genes, males had 3,685, and the p-values were more significant. So the males just have a greater magnitude of transcriptional remodeling. But the pathways are pretty similar. So they upregulate matrix genes, cytoskeletal genes, genes involved in cell adhesion. Interestingly, inflammation, there are a lot of genes upregulating inflammation in the males that we didn't see in the females. It's kind of interesting. That was an exception. And then a downregulation of energetics and mitochondrial genes, like quite profound. I'm showing 4 categories out of like 50 in the gene ontology analysis. So, and membrane transporters were the other things that were downregulated. So with exercise, the females, really, there was one gene that changed, which was pretty striking. And in the males, it was 3,500. And I really don't understand why the sex difference exists. But what we saw was a shift in most of these categories back towards non-transgenic. So in other words, the genes that were downregulated with disease are upregulated with training. So all the energetics, mitochondrial genes, and there's downregulation of matrix inflammation, cell adhesion. So we are seeing a partial reversal of the transcriptional remodeling in the male mice only. This is just another way of displaying it and showing, this is a Venn diagram where the pink, I know it's sort of hard to orient to this, but essentially the pink quadrants, or whatever these are called, segments here, are the genes that change in the opposite direction with HIT training compared to disease. So in other words, we're getting reversal about a third of the genes are going back in the opposite direction with the training. So again, I don't know whether it's the effects of exercise are set, the sex difference is that the exercise is having an effect only in males, or whether it's because the males are more severely affected that exercise is having a greater effect. And I don't understand that. And I think we're going to need to do a lot more studies to try and understand the reasoning behind that. So this is my concluding slide. So just to kind of bring you back to the landscape again, where we are now is that, we do have new therapies that we're able to employ in patients today. The myosin modulators for at least for obstructive HCM, and we will hope to maybe move that needle back to patients who are symptomatic with non-obstructive, and maybe ultimately even to those who are asymptomatic as a potential disease modifying drug. In terms of modulators of secondary modeling, we now have shown that Valsartan and antagonism, the angiotensin receptor, the angiotensin system is beneficial in terms of disease modification in early HCM. And I think that there are increasing numbers of studies that are now showing that lifestyle interventions, those that increase exercise, reducing body mass index, and controlling hypertension are really important in preventing the progression of disease and symptomatic HCM. So as far as things that are earlier, I think that we might be on the cusp of improving metabolics and energetics in patients, but we still have a very limited number of drugs that can do that. And eventually that's probably going to end up being kind of a combination of lifestyle factors and pharmacologic therapies. We and others are doing work in terms of looking at protein stabilizers, things that might stabilize a misfolded protein, or that might increase the stability of the wild type protein so that you can, maybe shift the balance and maybe reduce the amount of haploinsufficiency, for example, in those that cause loss of function. And then gene therapies, gene replacement therapy, is already here for diseases like Danon disease, muscular dystrophy, but still a lot of hurdles in terms of the adenoviral vector challenges with the dose, toxicity, et cetera, and specificity. But I hope, hopefully those will be overcome, and a lot of people are working in this area. Gene replacement therapy for myosin binding protein C mutations in HCM is likely to, be in clinical trials within the next year. So I think there's a lot of exciting things in the field, but we do have to keep in mind that obviously we're going to have a much greater opportunity to prevent disease if we start early, but we also have a very low risk tolerance for safety in treating patients. They're not even patients, like some people who haven't developed the disease or individuals who have a genetic predisposition, and may go on to live a perfectly normal, healthy life and be non-penetrant. So we don't want to harm anybody. And so that's why it's important that even things like gene replacement therapy, those trials are going to be done in patients with advanced disease to start, because, we can't sort of just give, viral vector gene replacement to people who don't have a disease yet. So it's going to take some time, but I think we'll eventually continue to work our way back to the left side of the curve. And so for me, it does always come back to patients. And I always, again, think about families when I'm thinking about, designing clinical studies and thinking about what's the right target and what should we really be thinking about. This is a family I've known for a long time. The father has had a really rough go with his HCM. He's had, he developed it when he was a child. His father died 2 years after he was diagnosed. He died suddenly. And he's had atrial fibrillation. He's had shocks for VT. He's been saved at least 3 times from sudden death from his defibrillator. He's gone on to raise 2 kids, go on to live his life to the fullest, and they travel and they do everything together despite, he's impaired at this point in terms of symptoms, but he's really valiantly charges forward. And and I think about his kids as I watch them grow up. They're actually right about the same age as my younger son. And, and I think about what their life may look like compared to his. And, it's already a little bit better. And I'm hoping that by the time they get to his age, that they're not going to suffer the same complications that he's had. So that's the end of my seminar, and I'm happy to take any questions.
They're really terrific. I don't want to take too much time. If you, with your permission, comment a clinical question
Yeah.
and then a science question, and I'll leave it at that.
Okay.
The comment is